Hi, welcome to Nourishable. I'm Dr. Lara. Today we are so lucky to be joined by Dr. Sarah Berry. Dr. Berry is one of the lead researchers at the PREDICT studies, which are these large-scale, enormous nutrition studies that are leading us towards personalized dietary guidance. And this is the research that has founded the ZOE nutrition product. So welcome, Dr. Berry.
Thanks, Lara, for having me.
What does PREDICT stand for, and what makes these studies so unique?
So PREDICT is actually a program of research, and it stands for Personalised Response to a Dietary Intervention Trial. And it's all around this area of precision nutrition. So looking at personalised responses and really unravelling what determines how each and individual one of us responds to food. And this is really where nutritional research is going. So, traditionally nutritional research, we've focused on the mean response. And traditionally, when we give advice, we give it at the population level to the average person. But we actually know that food is enormously complicated with thousands of chemicals, that we as individuals are enormously complicated, with our thousands of different kind of biochemical pathways. So throw the two of these together and you get huge variability in how each one of us responds to food. And so the PREDICT program was set up to really address kind of three key questions. So first was to really try and quantify how much variability is there really between individuals and how they respond to food. So how variable would your response be to exactly the same food compared to my response? And then the second thing we wanted to do was, can we actually look and unravel the determinants of what's causing this variability? So can we look at the multi-omic determinants, so genetics, metabolomics, metagenomics, our gut microbiome? Is it about just the food we eat, or is it about how we eat? Is it about time of day, how much sleep we've had? And then the third part was really for us to be able to use all of this information to build prediction algorithms using the expertise of ZOE, who are the startup tech company that are behind the PREDICT program, so that we could combine this, all of these findings from the PREDICT program together with this AI expertise to actually predict at an individual level how each person would respond to a given food.
Something that seems to really stand out about these PREDICT studies is that they're measuring the postprandial response as compared to the fasting responses, which is typical from many previous studies and as well as many clinical labs. Why is it important to look at the postprandial responses?
So I'm very biased because I spent the last 25 years of my research career studying postprandial metabolism. But I shall step back a little bit from my own passions on postprandial metabolism. Traditionally, you're totally right. If you're asked to go to the doctors, you're asked, fast overnight, we want to do a fasting glucose, we want to look at your fasting cholesterol levels. But that actually isn't the physiological state that our body is in most of the day. What we now know is that what's happening after we eat a food, which is called our postprandial metabolic response, is really important in terms of our disease risk, in terms of our metabolic health, in terms of our hunger, satiety, energy intake. And so if I could just explain very simply what happens when you consume food. So if you were to consume a mixed meal, so it's containing mixed nutrients, so fat, protein, carbohydrate, and fiber, what happens is that the glucose that's in the meal causes a short, sharp rise in blood sugar or blood glucose level, which we call postprandial glycemia. And it peaks around 30 minutes. It returns to baseline around 2 hours. What happens from the fat in the meal is you have an increase in circulating levels of what we call triglycerides, which is basically fat from the meal in the blood, that peaks around 4 hours after consuming the meal and returns to baseline maybe around 8 hours. Now, if you map those responses onto a typical eating pattern where you consume breakfast, lunch, and dinner, and maybe 2 or 3 snacks a day, which is a very typical eating pattern, that means that you spend most of your day in this kind of metabolic flux where you've got these short, sharp rises in glucose, you've got these prolonged rises in triglycerides. And so you spend about 18 hours of your day in this state of metabolic flux with either elevated glucose or elevated triglycerides from your meals. And the reason this is important is because we now know that these fluxes that you have are independent risk factors for cardiovascular disease, type 2 diabetes. They may also be associated with obesity. And we are also starting to understand the mechanisms. So we know that these peaks and these troughs, trigger oxidative stress, inflammation, remodeling of particular lipids that make them quite atherogenic, so are harmful in terms of your blood vessels. We know that if you're getting dips in your glucose, that you're going to feel more hungry later, that you increase your calorie intake, for example, at a subsequent meal. And so given that you spend nearly 18 hours of your day in this state, it actually makes sense that we look at these postprandial responses. And what we're really starting to understand in nutritional research is that it's these postprandial, which we often also use the term acute. So it's these acute postprandial responses that underpin many of the long-term, so what we often call the chronic, the long-term effects of foods on our health.
Right. It's so interesting because it is such an important exposure that it makes far more sense to look at what the state we're in most of the time.
I think it's important that we look at both. So, and this is what we're doing with the PREDICT studies. So we're looking very specifically at postprandial responses at both glucose and triglycerides, but also all of the different hundreds of metabolic markers that change postprandially. But we're also still looking at kind of baseline fasting values because they still give us another piece of the puzzle. And I think that's what's key here is that, you said earlier that what has impressed you about the PREDICT studies is the scale of them. We have thousands of people, but what for me as a nutritional research is really exciting is that we also have a great breadth of measures. We've got hundreds of different metabolites we look at, all of the gut microbiome, and we also have a really high precision and really high depth as well of data that we're looking at. And so what we're actually doing is collecting all of the pieces of the puzzle so that we can actually make a really clear, full picture of what's going on, after someone's eaten particular foods.
Right. And it really seems like everybody's puzzle is a little bit different.
100%. I mean, what struck me is something that I'd never really considered before I got involved in the PREDICT studies is the enormity of the difference. So we see more than a tenfold difference in how different individuals respond. So we might have two individuals who are a similar age, same sex, same BMI, but actually that person's response to maybe a bowl of pasta versus my response could be hugely different. And so what's right for them isn't going to necessarily be right for me. Yet on the surface, you might think, they're pretty similar in terms of, their physiology potentially, so they must respond the same.
Right. It's absolutely fascinating. Something else that I have noticed kind of within the nutrition fads and, on social media and all this is that there are a lot of direct-to-consumer genetic tests out there right now that are saying they can give you personalized dietary guidance based on your genes. What have the PREDICT studies shown about the influence of genes?
What we've been able to do, like I said, is tease apart all the different pieces of the puzzle. And genetics is one piece of the puzzle. It's important for some people, but for many of us, it doesn't have the big role that I think we previously thought. It's important just to mention as well that our PREDICT-1 study, which was the first study, and this is the study that was recently released in Nature Medicine, had about 70% of our participants were twins. So Tim Spector, who is the lead of the program of research, runs the UK Twins Registry. And so we were very fortunate that we had a huge amount of identical and non-identical twins, which really allowed us to tease apart how much of this response is due to our genes and how much is due to all of these other factors. And so what we were able to see is dependent on the particular outcome measure that we were looking at. So depending on whether it was the microbiome or was it postprandial lipemia or glycemia, we were able to quantify how much contribution genetics makes to that response to the food. So, for example, we found for postprandial lipemia, so our increase in blood fat after the following fat in a meal, that it had less than 5% contribution. So that actually genetics played almost no role. We found for glucose, it had about 30% contribution. So yes, it did play a role, but it still wasn't the biggest piece of the puzzle. And then what's I think really interesting is we looked at the microbiome composition in our twins in our identical twins, and then we also looked at the microbiome composition in our non-identical unrelated individuals. So people that weren't siblings, totally unrelated. And what we found is that identical twins share about 34% of their microbiome, and unrelated individuals share about 30%. So actually, their genetics is only causing that 4% increase. And so it's really showing that, yes, it does make a contribution, but actually it's tiny. We need to look at the bigger piece of the puzzle. So for all of these people that are going out there and buying these genetic tests that tell them what to eat, what day of the week to eat, how to eat, are really wasting their money if they're looking at that in isolation. We need to look at these other pieces of the puzzle.
So something that I think is, really relevant, especially right now. So here in the U.S., the new 2020 dietary guidelines just came out about a month ago. So these are population-based dietary guidance. And some of the really major findings of the PREDICT studies are that we really should be moving towards personalized dietary guidance. And so I was curious about what you think about whether there's still some role for population guidance, or how we could kind of move forward in making personalized guidance available and accessible to everybody?
Yeah, I think that's a really good question and a really important question, because as exciting as many of the developments are in nutritional research, at the heart of everything we do is always the thought of how can we apply this to the population and how can we apply this to individuals. And I think that to answer the first part of that question, I really believe that there is a place still for population guidelines. We're not yet, but I believe we will be in the future. We're not yet at a position to be able to give to every individual in the population, precision nutrition, personalized guidelines. And I think that, the kind of advice that we're giving with personalized nutrition is underpinned by health eating principles. And so population guidelines have their role alongside precision nutrition. I don't think one excludes the other. And what we do know is that we can really optimize within broad healthy eating guidelines at an individual level. So the health eating guidelines, so increase your fruits, increase your vegetables, reduce refined grains, increase whole grains, etc. All of these stand true, but it's the relative balance and it's the specific foods within groups, etc., that we can really personalize.
Right now, we know so many people aren't following the population-based guidance, for one reason or another. But then sometimes I also wonder if by getting personalized guidance, would that be a stronger motivator for behavior change?
Yeah, I think there's a couple of really relevant points there. So I think there's the whole point around why are population guidelines failing? Is it because they're wrong? Is it because people aren't following them? And we don't really have the answers for that. There is some evidence to show, certainly from work we've done at King's, that if you follow the UK guidelines, you have a big improvement in health outcomes versus if you stick to your typical diet. I don't necessarily endorse all of the guidelines. I think there's some things that could be tweaked, but I do believe that if we did follow them, we would see an improvement in health outcomes. The problem is that, again, they are at this population level. And so I also think there's an issue around a lot of mistrust around not just nutrition guidelines, but generally around what the public are told. One day, I saw this as the headline in my paper, and the next day they said this, and nutritional scientists have got it all wrong because they said we could eat this berry and now they're saying we should eat this. And that comes back to what I said at the beginning. The problem is that we're so complicated. Food is so complicated. And nutritional science is just advancing at such a rapid rate that it's not that we've got it wrong before, it's that we're finding that it's a bit more complicated than we originally thought. And I think thinking about the second part of the question about would actually people follow it, personalized recommendations more than population guidelines, that's key to this, because there's no point coming up with, this very high quality personalized advice if you're not going to follow it anyway. There's some good evidence from a rather large EU project called Food for Me, that shows that people do follow personalised dietary advice more than they would adhere to population average dietary advice. And this was a large project across different European countries where they randomly allocated participants to different levels of advice. They gave them genetic only advice, they gave them precision nutrition advice, and then they gave them population average based advice. Those individuals that were given personalised nutrition advice were those that were most adherent and made the most beneficial changes to their diet, showing that actually it does probably facilitate greater adherence to the dietary recommendations.
Mm-hmm. Yeah, it's such an interesting question. Once we factor in behavior change, that's such another layer of complexity into our big jigsaw puzzle.
And I think that's really important that as nutritionists, when we're giving advice, that we have to understand that there's so much more to this than just the food we eat. There's the social, there's the cultural, there's the emotional aspects around food. And many of us don't eat alone. We're eating with our families, or we're eating because, we're fed up like me of homeschooling and, and juggling everything, or we're tired, or, there's all of these other aspects, or we have this cultural, preference, etc. And so I think something we need to be very mindful of as nutritionists when we're developing any kind of recommendation, but particularly at personal levels, how does this fit in within those behavioural constraints? And that's the kind of thing we're trying to do very much with Predict, that we're trying to make sure there's no kind of exclusivity, and that we're not excluding foods. We're really pointing people in the direction, empowering them to be able to choose within a whole selection of foods that are best for them.
Mm-hmm. And that's actually really interesting that you bring that up. So today is my first day on my 4-week ZOE nutrition plan. So I've spent the past few days, planning and grocery shopping and using the food scores. And I have felt that kind of empowered feeling of I'm not being prescribed something.
Yeah.
I'm being, the choices, the best choices are being shared with me. And so far I've really appreciated that. So far I've had breakfast, but we're off to a good start. But just building on the ZOE nutrition plan. So I've been, as I said, have started using this ZOE Insights app, which provides me with scores for all these different foods based on my personalized responses, as well as this machine learning from thousands of other participants. Tell me a little bit more about how these different foods get their scores.
Okay, so the foods are allocated scores based on 3 factors. So they're based on the microbiome, so the gut score. They're based on a glucose sugar score, and then also on a fat score. So firstly, they're personalized to you. So before you started the ZOE program, you undertook a testing period. So you would have consumed our beautiful muffins that are specifically designed to stimulate quite a pronounced glycemic and lipemic response, so a pronounced postprandial increase in glucose and in triglycerides, so that we can measure your own unique blood fat and blood sugar control. You also provided us with a stool sample so that we could look at your microbiome and look at your own unique composition of your microbiome. So what specific good bugs you have, what specific bad bugs you have, and look at them in relation to your current diet as well. And then by integrating those findings from you as an individual together with what we've been learning from our Predict 1 study and our Predict 2 study in a couple of thousand people, and also a wealth of scientific knowledge from all the experts like myself and Tim and many other experts working on the Predict program, we've generated scores across those 3 themes, so across your gut, your sugar, and your fat control. And so then for each individual food, we can give you a personalized score based on what we know is in that food and how we know you will metabolize that food.
Right. It's fascinating. And it's also been very surprising to me. Sometimes I would, use the app and scan a barcode of a food that I thought was really healthy. Like I have a particular favorite muesli that no added sugar and that I thought was great, and it got a really low score for me. So I can already see how these food scores are going to help kind of shift my dietary pattern, which I thought was really healthy based on, my previous knowledge of nutrition.
Yeah, and I think, something we're trying to achieve is shifts, like you've said. So we're not trying to radically change your diet, because that's not going to work. If you want muesli and that's what you have most mornings, then it's just a case of saying, okay, this muesli is slightly better for you because you have better fat control than sugar control, for example, and therefore this is the best for your own unique biology.
Mm-hmm, absolutely. I have to ask about the muffins. So I understand you created these muffins.
Yes.
Yes. It was very interesting eating them. They're definitely sweeter than what I typically would eat for breakfast and lunch, as I did on the ZOE test days. Tell me a little bit about the story of creating these muffins. What was that like?
So these muffins were created about 20 years ago when I first started researching in postprandial metabolism. So during my PhD, I started undertaking randomized controlled trials in healthy humans, looking at how different people respond to different types of fat. And I spent many months in our dietetics kitchen at King's College London trying to devise some kind of single food that we could give to people that would deliver a certain amount of fat and a certain amount of carbohydrate in a very controlled way. And I tried everything from biscuits, all sorts of kind of savory foods. And muffins was actually the only individual food item that we were able to really play around with the composition. So we were able to incorporate large amounts of fat, large amounts of carbohydrates, but also manipulate the relative amounts. So on the PREDICT study, what we're interested in looking at is not just one given amount of fat and carbohydrate, protein and fiber, but we want to give a whole variety of different proportions of these nutrients. And within muffins, we can deliver a muffin that has, for example, 50 grams of fat, 85 grams of carbohydrate, down to a muffin that might have almost no fat and be exclusively sugar, and all within a very refined kind of food vehicle. And that's quite important because we're really starting to understand the importance of the food matrix. And by that, I mean kind of the structure within which a food is. And so if I was to give to you the same amount of nutrients in a different kind of matrix, so if I was to give it to you as a mixture of, let's say, nuts and some other plant foods, etc., it would have a highly variable impact on your postprandial responses because cell walls come into play, the fiber comes into play, how much you chew the food comes into play. So the idea was, as well as being able to incorporate quite a range of nutrients, was to be able to actually give it to you in a really simplistic, refined way that there's no added complexity of matrix as well.
Very interesting. And that actually really brings me to my next question, which is that there has been lots of attention on ultra-processed foods. And I noticed, especially in the microbiome paper, that there was some analysis about ultra-processed versus minimally processed foods. What have the PREDICT studies shown us about the impact of ultra-processed foods?
So in the PREDICT study, we've categorized how we've looked at our responses to food at kind of 4 different tiers, which is really, again, what we should be doing in all nutritional studies. So we've looked at the impact of nutrients, we've looked at the impact of food items, we've looked at food groups, and then we've looked at kind of dietary patterns, so dietary indices. And what we can do is, with also the food items that we've looked at, we can categorize them into healthy, unhealthy, plant, into highly refined, highly processed, and unprocessed. And we find that you get, in terms of the microbiome, a really clear separation of what we're calling these good bugs and these bad bugs according to the quality of the food, according to whether it's a healthy or unhealthy plant, and whether it's highly processed or unprocessed. So you might have something like white bread, for example, which is a highly processed grain, and that is heavily associated with these bad bugs. Yet you'll have a very similar food, but it would be whole grain bread, which is less processed. Obviously, there's still some processing. It's less processed, and that's associated with the good bugs in our microbiome. And we see exactly the same separation when we're looking at postprandial glycemic responses. I mean, I don't think there's anything new there, but we know that heavily processed foods is more bioaccessible, so the nutrients are more available for rapid absorption and digestion. So therefore, you have this bigger peak in glucose and then a bigger dip as well.
Right. And to me, the findings about the ultra-processed foods really seems to be one of the big take-home messages that can be perhaps applicable to everybody outside of the personalized nutrition responses.
Yeah, I mean, I think, although very little direct research has been done in terms of randomized control trials, apart from the work by Kevin Hall's group, I think it's becoming very evident across all kind of disciplines in nutrition of just the importance of having this highly bioaccessible, refined, processed foods versus trying to keep foods within their original matrix.
Mm-hmm. Right. The food matrix matters.
Yes, definitely.
So I am an avid coffee drinker, and I was very interested in the associations found between the microbiome and coffee. Can you tell me a little bit more about what the PREDICT studies found regarding coffee?
Yeah, so we found that coffee was associated with a very specific bug. So we could almost predict coffee drinkers based on the presence or absence of this particular bug, which was quite exciting. And we find, out of all of the foods that had the strongest association with one single bug, a lot of the other foods were associated with more groups of bugs. But we could potentially take two individuals, a coffee drinker and a non-coffee drinker, and potentially identify them based on this single bug. But importantly, this bug was part of our signature of good bugs, and so was heavily grouped in this healthy microbiome signature.
Oh, that's fascinating. Well, it definitely makes me feel good about drinking my morning coffee. Oh, something that I thought was so interesting was that the gut microbiome seemed to be a better predictor of the postprandial triglyceride responses compared to glucose. That seemed to be one of the findings from the microbiome paper. Why do you think that is? That seems like to me, it surprised me, but perhaps you have a deeper insight about why there might be that association.
No, we don't know. We were surprised as well. So a previous group has looked at the relationship between the microbiome and postprandial glycemia. This is led by Erin Segal's group in Israel. We found similar findings to his group in terms of the relationship between the microbiome and glycemia, but we're the first people to ever look at the relationship between the microbiome and lipemia, so this postprandial increase in triglycerides. So we didn't know what we were going to find. What, like you said, was really quite exciting is this really strength of the association, the fact that it was even stronger than this relationship with glycemia. It's likely it might be due to the way that fat's metabolized and the relationship with bile acids, that, the microbiome is involved in processes around that. But it's something we don't have really clear answers to yet.
Right. So interesting. The other thing that I thought was really impressive was that in the microbiome paper, it seemed like the findings were very consistent, that there was this group of, 15 good bugs, 15 bad bugs identified, and they were consistently, the good ones were consistently associated with good cardiometabolic health, obesity indices, healthy dietary pattern indices. Do you feel like these PREDICT studies have kind of defined a healthy microbiome, or is there still a lot of diversity of different healthy microbiomes out there?
I think that we're still in the early stages of our discoveries on the microbiome. And so I think that it would be premature to say this is the definitive healthy and non-healthy microbiome. Based on what we know and based on the quite advanced computational biology that's been used in this study, I think that what we have found is very cutting edge. I think that it's reliable. I think that it's exciting that this is the first study that's looked at the relationship between the microbiome and diet and the microbiome and health measures within the same study. And like you'd said, the consistency that these 15 good and these 15 bad bugs were consistently associated with the diet, with all of these different health measures. I think that I am quite confident that this signature that we've developed, certainly based on current developments in the microbiome, is, a good signature of microbiome quality for these good and these bad bugs.
Have you done the ZOE test? And did you find that did it change how you eat?
So I have to confess I haven't done the ZOE test. I think that I'm the only person at ZOE that hasn't done the ZOE test. So I've been running postprandial studies for over 20 years now. And at King's College, we've always been very careful about self-testing, and we've been rather strict on certain ethical considerations regarding coercion of for you or your colleagues being coerced into doing their study. So no, I haven't. And I would love to.
Well, that does sound like a very responsible thing to do, though. So I completely understand that. Yeah, so I'm starting my 4 weeks that we planned today, and something that I'm already curious about is finding out whether these changes in my dietary pattern are going to ultimately change some of my metabolic responses. And when I was scored as a poor responder to fat, so that's something that I'm very motivated to try and fix, because it was a big surprise to me, and it was something that had never been noticed from any of the fasting samples that had been taken before. So is there anything in the works with the PREDICT studies or with the ZOE test where we could kind of do a retest after having consumed the personalized diet for a while?
Yeah, so in terms of us validating our advice, we are going to embark very soon on a randomized controlled trial where we will look at the efficacy of how pre and then post, like you say, people's metabolic responses are. We're currently we're always evolving the ZOE product and our investigations, and that's what makes not just ZOE but the PREDICT research so exciting, that it's not a stagnant product, that it's evolving as we're making new discoveries. One thing that we're currently discussing is exactly what you said, that seeing if maybe at the end of a 6-month period or 3-month period, or at a certain time period, we can offer people to do a retest. It's certainly something we'd like to do with the microbiome, to see if there's a modification of the microbiome, and we're currently discussing, would it make sense as well to be able to offer that they can retest their metabolic control. So like you said, see if actually it's improved your fat control, so that hopefully you're now a good fat responder. So it's all I can say is watch this space. And I think it's going to be like this over the next year, which I think is actually quite exciting, because we're learning from our own research at a really fast pace. And if we can push that back into the actual ZOE product as well at the same time, is a great way that people can benefit in real time from our exciting findings.
Right. I think so far the experience I've had is that it makes me feel very engaged in my health and in my food decisions, which is definitely a great way to motivate sustained behavior change and develop new behavior habits. What do you feel like you're excited to see as this research moves forward?
We're sitting on a gold mine of data already from the PREDICT-1 and the PREDICT-2 study. So as a nutritional scientist, I'm super excited to delve deeper into the data that we have. And again, that's what's really exciting with these studies, and the collaboration we have with the tech company and the machine learning, data science expertise, that at quite a rapid pace, we can start to really delve into all of these different questions. And in real time, as new discoveries are made, we can then answer, the next question. What's really exciting as well is that we have a PREDICT study embedded within the ZOE product called PREDICT-3. And so we're really capitalizing on citizen science, which I think is a great way forward, because we're benefiting the customers for ZOE, but we're hugely benefiting nutritional research and precision medicine. And I think, once we get this vast amount of data again at this depth, this scale, this precision, I think, we're just going to be able to answer so many questions that we have just never even had the ability to answer at this kind of depth.
Well, I am very excited to be one of your citizen scientists because I contribute. I'm part of Predict 3. But yeah, I'm just so excited about this research and seeing where it's going. And I think it really has a potential to be, to really impact public health. So I think that is so exciting.
Thank you. And that's our aspiration. And I think what's great, like you say, is that everyone who's part of ZOE is also part of advancing science and advancing nutritional research, which is the best partnership possible.
Absolutely. Well, thank you so much for taking the time to speak with me, Dr. Sarah Berry. This was wonderful.
Pleasure. Thank you for having me.
Yeah. And as I said, I will, I look forward to seeing my continued experience with the ZOE test and just seeing where the research moves forward.
Great. Thanks. Lovely to meet you.
You too. Take care.
Okay.
Bye.
Bye.
Thanks for tuning in to Nourishable. Check out the video description for references to all the studies we talked about, as well as a link to my video review of the ZOE product, and be sure to subscribe to stay up to date on all things nutrition.